Hi there, everybody. Welcome to our next session in day one of our Genetic Evolution Revolution Summit. You're in the session for the late preclinical and scale up. So I hope you all enjoyed the presentation so far. I hope you had a chance to attend the discovery preclinical session with Kira Marshall and Angela Zhang. They demonstrated how easy it can be to get started on your genetic medicine drug discovery and development using the Spark, the Ignite, and GenBoy. And one thing I hope is really clear is that developing your genetic medicine using the PNI nanoassembler family of instruments really enables you to begin with the end in mind. Meaning, as you determine your ideal nanomedicine candidate using Spark, optimize the formulation and assembly parameters on Ignite, you know that there's a clear path for your candidate to the clinic, that our systems are designed to work together to enable the scaling of genetic medicine from lab scale to manufacturing scale. And this is made possible through the use of that same microfluidics architecture you just heard about if you were able to attend the session with Tim and Shell, with the same ability to finely tune critical parameters and through the modeling of critical unit operations, upstream, downstream processing on each of our systems from small scale to large scale. And so in this session, we're going to take some time to show you how you can now scale up your genetic medicine candidate to clinical scale, how you can perform process development activities in-house and get ready for your clinical batches, all while minimizing the risk of batch failure, minimizing the cost of each run, and ultimately accelerating the time to reach the clinic. And so I'm going to spend the first half or so to talk about the next system in our nanoassembler family, which is known as the Blaze. And then I'm going to pass it on to my colleague, Maria Karen, who's our lead engineer, who's going to provide a hands-on demonstration of the Blaze system, which includes our newly launched Blaze Plus feature, which allows the connection of external vessels and formulation of volumes of up to 10 liters. So as you've heard us describe, the mission of PNI is to help create transformative medicines, to create the genetic medicines, the therapeutics, the vaccines that will define the future of medicine. And now we're moving to the forefront of the genetic medicine revolution by providing that full stack genetic medicine product development with our proprietary drug product manufacturing platform, delivery platforms, as well as the expertise, education, and co-development programs. So in this session, we're going to be focused a lot on the platforms and in particular, the nanoassembler Blaze. But before I do that, let me show you that full nanoassembler family of instruments and show you how they're used together along that development pipeline. So to develop and commercialize a new genetic medicine, there's a lengthy drug development process, which is shown to be much shorter all the time, but still involves not only the discovery and development of effective formulations, but also the scaling and manufacturing of that drug product. And a lot of attention is being paid now to the importance of the consideration of manufacturability, particularly with respect to scaling of drug formulations during the earliest stages of drug development, and genetic medicine is no exception. So the nanoassembler family of instruments is designed with that end in mind, to work within the genetic medicine development framework with the end goal of enabling clients to develop and commercialize a nanomedicine drug product that's effective, safe, and manufacturable. So each of these systems has a distinct value within that framework, whether it's fast and efficient screening with the Spark, whether it's formulation optimization with the Ignite, large-scale studies and process development that I'll describe with the Blaze, and later on you're going to hear about the clinical and commercial production by our GMP system. And moreover, they're designed to sort of work together to enable the smooth transition from one system to the next. And so the more smooth and efficient that transition is, the quicker the drug product can get to market. And the way that this is facilitated is, of course, by using this proprietary next-gen microfluidics manufacturing technology, gives you the robust and reproducible production of uniform, high-quality nanomedicine drug product. And the other part that's important is, of course, you can model the same critical unit operations using all of our systems, which allows clients to develop a robust, scalable process from the earliest stage, simplifying scaling and reducing the need for extensive re-optimization on higher scales, which ultimately saves in material and labor costs. I'm going to show this unit operations in the next slide. But you heard about Spark and Ignite in the previous sessions, again, providing very, very simple way and fast way to develop a nanomedicine drug candidate to identify the critical quality attributes, the CQA, and to determine some of the critical process parameters, the CPP. And you can perform a lot of your preclinical testing in vitro and small scale animal studies. And so now you're ready to take the next step. You're ready to scale up your formulations, and this is going to take place on the NanoAssembler Blaze. The Blaze is designed to facilitate and accelerate program advancement. It processes scales from 10 mils to 10 liters and allows clients to begin optimizing at a clinical scale. So this unit operation, I hope you've seen it before, it really is important to sort of emphasize that we are not really just talking about the process of the nanoparticle assembly. We've created this system that enables you to model and scale the entire unit operations using our NanoAssembler platforms from Spark to Ignite to Blaze to GMP. Because we know that at later stages, if you're to bring in these other critical unit operations at a later stage, when of course the volumes are greater, and of course you're at a more critical phase of the drug development pathway. So if you end up with a failed batch, it can step back or even derail the program. So the Blaze, again, as is all of our systems, allows modeling of the complete unit operations using an RUO system. So now this can be done at a higher scale than the Ignite, but again, it's still at a lower cost per run than running this on the larger GMP system. So ultimately, this reduces the overall cost of the program. Again, you reduce the need for extensive re-optimization of those critical parameters at scale on the more expensive GMP system by performing them on the Blaze. So before I tell you all about the Blaze system, I wanted to give you a bit of background, but now I just want to talk a little bit about some of the common issues that are faced when scaling a drug candidate. So I think everybody here is aware, or should be aware, that really one of the most critical aspects of a drug development program is the manufacturability and the scale up. And of course, we talk to a lot of clients and customers and drug companies about some of their major pain points. And again, the most common theme we hear from drug development companies is that the process of scaling is both expensive and has a higher level of risk. And so one of the major benefits of using nanomedicine as a drug delivery method for genetic medicines is that you already can operate on a much smaller scale than traditional viral vector-based delivery systems, which can not only be beneficial from a cost and a footprint perspective, but it also means your final manufacturing scale is smaller. So therefore, less optimization may be required. However, nanomedicine is not immune to scaling considerations. No matter what, eventually you're going to have to do that large scale experiment, and it's going to use a large amount of reagents, and it's going to be expensive. Now, the reason that we describe it, or we hear it's called higher risk and expensive, it's really two main reasons. So number one is the obvious reason we all think about, which is these various processes and reagents and systems, they don't behave the same way at large scale as they do at small scale. So when you scale up your formulations towards manufacturing scale, you need to make sure your critical quality attributes, your CQA, are able to be achieved when you're working at higher volumes, higher flow rates, longer run times. And so the main way to mitigate this risk is really to optimize those CPP at smaller scales and try to predict how it'll behave at larger scale. So if you can, minimize any changes to those CPP that'll occur at larger scale. But there's only so much really you can do. We enable you to do as much as possible at those smaller scales. There's another reason, though, that scaling can introduce risk to your program, and that's because when you need to perform high volume runs, it often needs to take place in another facility. So in many cases, large-scale equipment that's required to perform those large-scale runs is often the same GMP manufacturing equipment that's used for clinical and commercial production. And that means it's almost certainly performed by a contract organization such as a CDMO. So when the large-scale studies, the ones you're doing that are particularly expensive, are performed in a different facility, you also have to ensure that all of your protocols, all your SOPs have been clearly communicated to that external organization outside your company. And it's not uncommon to learn that even the best written SOPs and even the clearest work instructions can be performed differently in different facilities. So it can be a much less risky process if you can perform process development in-house, where there's close communication between your development, your tech dev and your process development teams. The lines of communication are more open, they're simpler, they're quicker. So at Precision, we aim to minimize the risk and the expense of scaling up two different ways. One is by using that same next-gen microfluidics technology that gives you reproducible, highly controlled particle formulation at every scale, and use the identical unit operations to ensure that the upstream, downstream processing steps can also be modeled at scale. And the second reason is our system, the Blaze, for performing these large-scale formulations, enables you to bring almost all of your process development activities in-house. So our goal is really, when you need to perform that really expensive run, meaning you need to use a lot of expensive reagents, that chance of failure is minimized. Another big advantage, I'll just finish off here, of running your development in-house, you can avoid a lot of the scheduling issues and bottlenecks that are common with CDMOs, especially as the demand for these facilities has recently skyrocketed. So when you control your own process development, you own your own timelines, you can move more quickly through the drug development pathway. Okay, so let me introduce you to the Blaze system. So the Blaze system is our large-scale preclinical system that's designed to facilitate program advancement. So when you're ready to take your drug candidate towards manufacturing scale, when you're ready to begin process development, it's time to move to the Blaze system. Blaze is designed for later stages of preclinical drug development within the drug development framework, studies that require larger volumes and higher flow rates. Blaze is used to define the manufacturing process, to determine the CPP at scale, and to de-risk the eventual transfer of your manufacturing protocols over to the NanoAssembler GMP system. And it can all take place on a lower cost system. So how does it do that? Well, as I mentioned, the Blaze system uses that identical microfluidics cartridge. It uses the same technology, but in particular, the Blaze uses the identical microfluidics cartridge as the one used on the GMP system, which really is the most novel and critical component of the nanoassembly process. So the use of identical cartridges on the Blaze allows clients to perform most, if not all, of the essential process development studies using a lower cost RUO system, which minimizes the risk of failure when you transfer to GMP. So essentially, when you're transferring your protocol over to the GMP, all of the CPP have been determined on the exact same microfluidics cartridge that we used on the GMP system. So there shouldn't be any surprises when you transfer over to GMP. And the other value that I talked about is that it enables clients to perform process development activities in-house, retaining control over process development rather than outsourcing, which minimizes risk of failure and accelerates your drug development pipelines. And it allows a very smooth transition from small-scale system to the Ignite, all the way to a clinical production system. All performed in-house, all performed in the same location, simplifying protocols, transfer of information, and progress to the clinic. So now I'm going to show you how Blaze can be used to scale up formulations from Ignite and transfer those optimized protocols over to GMP. Okay, so for simplicity's sake, I'm not showing the Spark here. It's quite a small scale, so it actually falls off the left side. Here's how the drug programs can scale from Ignite to Blaze to GMP. So Ignite here will perform formulations up to 20 mils in volume, up to 20 mils per minute. You saw a nice demonstration earlier today, how quick, how simple that is, done within single-use syringes. So when you've determined your CQA for your particle, you've identified your lead candidates, your optimized formulation, now you're ready to take the next step over to Blaze and start scaling up. As I mentioned, of course, it uses the same microfluidics technology, so there's a very nice clean transfer from Ignite over to Blaze. You're essentially going to be moving from one size of that cartridge, that next-gen cartridge, to the next size up. That next size up allows for higher flow rate, so you can start to make larger volumes in a nice amount of time. So the cartridges used on the Blaze are called the 400 and 500. I'll describe those a little bit more in detail. Again, identical to the ones being used on the GMP system. So your dimensions, your total flow rates have changed. And so it allows the manufacturing of larger volumes. And you need to optimize your CPP on this new cartridge. Now the Blaze can run formulations of up to 10 liters. And that uses this new Blaze Plus feature, which in fact we're launching today. I'm going to talk a little bit more about it later, and you're going to see a demonstration later. Essentially what this does is it allows the Blaze system to connect to external vessels. So once you get above 1 liter, it's pretty hard to contain that inside a sort of decent sized instrument. You need to start using external connections and connect up to external bioprocess bags. And so that can be done with the Blaze, another great way to model factors that you'll be ultimately using in GMP. So it goes all the way up to 10 liters. That's a very significant amount. Just to give you an idea how much of an mRNA LNP formulation that would be if you had a standard concentration of 0.1 mg per mil, that would work out to about a gram of mRNA. The flow rate goes up to 115 mils per minute. Ideally, this max flow rate will be sufficient to hit the limit size flow rate of the LNP formulations, such that the size of the particle is no longer affected by higher and higher flow rates. So when you move to GMP and you can start to expand or extend that flow rate up to 200 mils per minute, you don't see any changes to your particle sizes. So again, once you've performed all your process development activities on Blaze as much as possible within the specification of the Blaze, you're ready to transfer it to the GMP system. When you do that, you're using the exact same cartridge, same dimensions of the micromixer, no surprises. Now you simply need to perform runs at clinical scale, so volumes beyond 10 liters. You can go up to 50 liters for a validated method, and even beyond 200 mils per minute is our sort of max run rate. But again, options to go beyond that as we work with you on your manufacturing requirements. So again, ideally, you've completed the majority of your process development activities on the Blaze. You've done it at lower costs, which reduces the overall cost of the program. You've minimized failure of these large-scale clinical batches because you've optimized all the critical process parameters on that lower cost system using that identical microfluidics architecture. Using the Blaze Plus, again, even more of those activities can be done in-house on that preclinical system. Again, overall reducing cost, reducing time, and minimizing risk. So as I talk about a lot, I talk about how you can bring the Blaze in-house. It's not just, I talk a lot about the process development activities that are going to be performed, because ultimately that's the greatest value of Blaze. But it's not just designed to perform PD activities. Of course, any study where you need larger volumes in a single batch can be done on Blaze. So it can also be used for late stage therapeutic development activities. So anything that requires a large amount of material. Of course, when you're developing a drug, you must show it to be both potent and non-toxic in relevant models. So if you're doing these late stage, large volume preclinical studies, such as large cohort rodent studies, larger animal models, such as ferrets or non-human primates, you're doing second species studies, and you can do a fair number of toxicology studies as well, using the Blaze system, giving you large volumes in a single batch and allowing you to maximize your utility of the instrument by performing a variety of studies. And of course, the process development activities, which I've talked a lot about, these are studies that define, establish, and implement the manufacturing process. Let's have a little closer look at the Blaze. I want to show you some of the specifications. As I mentioned, the key is that formulation volumes can go all the way up to 10 liters. When you move between 1 liter and 10 liters, you're going to need to be able to connect external vessels. And for that, you need to have the Blaze Plus feature. Higher flow rates up to 115 mils per minute, a very simple and easy to use interface on a laptop, which you'll see in the demo. And again, allows you to perform all of these in-house using common lab equipment. The cartridges that are used on the Blaze, as I mentioned before, I refer to them as 400, 500. They're identical as the ones that are used on the GMP system. We also have a classic cartridge that can be used on Blaze. I'm just going to focus on the next gen cartridges because we're talking about that scalability and transfer over to the GMP system. It comes in two versions, the 400 and 500. Those refer to the relative dimensions of the channel. The next gen 400 has more of a dynamic range. It has a lower max flow rate, so you can get to your limit size sooner. However, that does lead to higher pressure and optimal use of this next gen for applications that don't require large batch sizes. So perhaps something more in the realm of personalized medicine, where you just want one dose per batch and you don't need particularly high throughputs. Also, your formulation may dictate this cartridge as well. The next gen 500 is best at higher flow rates. It allows faster runs. And so this Blaze should be used for defining that manufacturing process so that when you're ready to scale, you can take either the 400 or the 500 and run them on the GMP. They come in both the standard version or they have a version that has a dilution feature as well. Okay, now, I'm going to talk a little bit about Blaze Plus. We're very excited today because today is our launch day for our Blaze Plus. Essentially what the Blaze Plus is, as I mentioned, it really expands the capabilities of our Blaze system. So it's a functional mode of the Blaze system that really unlocks the ability of the Blaze to process nanoparticle assembly volumes of greater than 1 liter. So again, the existing capabilities of Blaze, the pumps, the cartridges, everything is the same. The Blaze Plus feature really allows you to connect those external vessels, those external bioprospects, gives you a chance to evaluate them on a lower cost system. So what that means is that you can perform more of your process development activities in-house. So prior to having the Blaze Plus, once you get over volumes of 1 liter, you need to start move to our higher volume system, which is of course our GMP system. We do have the ability to run that in an RUO mode, but ultimately that system is a more expensive system. The runs are more expensive and they're quite a bit longer as well. When you can do it on the Blaze, you save a lot of time, you can save a lot of money, and now you can go up to 10 liters. And so essentially you minimize that cost of transfer. Again, these activities need to be performed. When you can perform those same activities on a lower cost system, again, you reduce the cost of the system, you reduce the cost of the program. Again, you bring those things in-house, you're performing these in-house, the timelines are shortened. It's particularly good for accelerating your drug program. It's important to note that there's no difference between the Blaze and the Blaze Plus. So when you hear us talking about the Blaze Plus, we're talking about the same system that has that upgrade, which is a very simple hardware upgrade that adjusts the Blaze. It's got a software. The software will enable Blaze Plus, and we'll just show you exactly how to set up that external run. It's very simple to do, and so Maria is going to show you that next. Again, I just really want to highlight when you can perform more of the process development activities in-house, you can retain control of that PD process. Again, scaling is one of the more expensive and risky stages of drug development simply due to the sheer volume of reagents that you're using. So again, maintaining control of that process by keeping things in-house, you can better align your development activities with your PD activities before you end up having to outsource to a CDMO. Again, this reduces the cost, and I'm doing a comparison here because I really want you to see the difference between what it costs to run on the Blaze versus the GMP. There's a significant cost difference, not really in the cost of the fluid path, although the GMP is more expensive, it's a more expensive system. but it's really around the holdup volume. When you run it on the GMP system, there is a holdup volume of 100 milliliters. In the Blaze system, you don't have any holdup volume. You're able to recover all the fluid from the fluid path. And this is important. We made sure this is available to labs and clients when they're running these activities. Again, saving on reagents is critical. So again, if you're running a standard mRNA, 100 mils of a 0.1 mg per mil system can be around $25,000. So it's a significant price saving. Every time you can run something on the Blaze instead of the GMP, you can save about $35,000 US. Again, this is an average price, but I'm trying to use it to emphasize that there's a very significant cost saving when you can run it on the Blaze. It also saves a lot of time. So not only, again, I talked about how things can move smoother and faster, but the runtime on the Blaze is also much lower. So the GMP system does take about 2 full days to run the protocols. Of course, they're very standard SOPs that require flushing and cleaning and priming and calibrating an operation. You don't need to do that extensively on the Blaze. It's a much simpler process. Lower volumes can be run in as little as an hour or 90 minutes. So again, when you add this up over a number of process development activities, things can happen a lot more quickly on the Blaze, and that can have an impact on your program timelines. Okay, so the last thing I'll talk about, I just really want to show you, you've seen this before, I believe Kira and Angela touched on this data that we have that really show how the scalability or the performance of the nanoassembler systems is consistent across multiple scales and multiple flow rates. So as you recall, we encoded human EPO and mRNA. We packaged it into an LNP using our Genvoy, ILM, and NextGen technology. Again, that LNP is delivered to liver cells with express EPO, which stimulate the red blood cell production. And you can see both in the left column that essentially you're getting really similar and consistent sizing of particles, whether you're running on Ignite at 12 mils per minute. The middle two are on Blaze at either 60 or 90 mils per minute. And you can see when you transfer that over to GMP and run it at a higher flow rate of 200 mils per minute, again, you get this really nice, consistent sizing of particles with a really sort of low polydispersity index. So this system really shows that that scalability is, reproducibility is really retained all across scales. And from a performance perspective, you can see you can get an increase in the red blood cell production in mice. Again, consistent results across the scale. Here's Ignite at 12 mils per minute. Here are the two Blaze runs, 60 and 90, and the GMP at 200. So essentially, that EPO encoded mRNA LNP batch size was scaled by over 80 times using a single mixer configuration, minimal process optimization without any changes to the particle characteristics and biological performance. So I'm at the end of my section. I know we're excited to see what this actually looks like, and you've heard me describing the benefits, really. I just want to leave by emphasizing that this nanoassembler system really is transforming medicine, particularly genetic medicine, and especially in this day and age. This year in particular, we've seen a huge amount of increase in adoption and, of course, interest in this. It is a really groundbreaking technology that enables us to develop those genetic medicines sooner. You can see in a number of publications. I urge you to go to our website to have a look at the various publications and resources. But as we start to, of course, see more and more of our clients getting excellent results and making clinical batches and using our systems to actually develop clinical and commercial batches of LNPs, we're really excited to be able to share that with you. So keep coming back and having a look. And so that's the end of my section. I'm going to pass it on now to Maria, who's going to demonstrate how our Blaze and our Blaze Plus works. So Maria.
And I'm the lead engineer on the Blaze system. So I'm here today to give you a bit of a demo on the Blaze, and in order to do that, I'm going to talk through the various components on our fluid path that are used for optimum flow conditions in order to formulate on our system. We will then set up a recipe and run a demo using the water loaded on the blaze here. And then lastly, we're going to set up the blaze for a Blaze Plus run. So I'm going to show you how to attach the tubing that comes in the Blaze Plus tubing kit and how to attach the external vessels so that will facilitate a large batch run. So let's get started. So the first thing is the next gen cartridge that Vicky described. So this is our next gen 500 cartridge, and these cartridges slot into the front end on the Blaze system. So there we go. Once we start a formulation, the first thing that happens is the sippers come down into the vessels that are loaded onto the base of the blaze. The pumps here then open and close to draw fluid into the fluid path and then push the fluid out towards the cartridge. On the way to the cartridge, it passes through a pressure transducer, and there the pressure of the system is monitored, and this allows for a diagnostic tool and also for overpressure protection on our system. So the fluid enters the cartridge here, and this is where the mixing occurs. It then exits the cartridge through the spout here, and we have a automated sample collection or waste collection arm that moves in and out. So initially we can collect some of our start waste, and then after the set length of time, the arm retracts back and allows the collection of the sample into a vessel. And there's various configurations for different vessel sizes here. So that's pretty much the main components on the fluid path to allow for optimum flow conditions. So now we will move over to the laptop screen, and I'm going to show you how to set up a recipe for a run that we are about to perform. So once you get to the Blaze software, the first thing to do is to create a new recipe. So we'll click the new button and let's name it Demo Blaze. So in the drop-down menu, we select the cartridge type that's been used, and because we're using a next-gen 500 cartridge, we're doing a high flow with dilution. We now specify the volume, and for today's demo, we're going to do a 20 ml formulation at a 3:1 reagent ratio, and we're going to do a flow rate of 60 ml per minute. As Vicky mentioned, the maximum flow rate is 115 ml per minute. We will keep a dilution ratio of 1:1, and we will keep the sippers on. So I'm going to describe later why we would turn the sippers off when we're setting up for Blaze Plus. We will set the start waste to 4 ml and the end waste to 0.5. And our user guide details how to specify or how to select the best start and end waste for the system. So we'll then save that recipe, and then we are ready to click Formulate, and that will bring us onto a second screen, which gives us a list of checks to perform. And the first one here is to check that the formulation parameters on the right are correct. So this table here gives us a breakout of the parameters that we inputted when we created the recipe. You can see we have a flow rate ratio of 3:1, our volume is 20 ml, and our flow rate is 60 ml per minute. We have a dilution ratio of 1:1, and so it breaks out the start waste so we can understand how much extra fluid we need to load for the initial and end waste. It also gives us it gives us the length of time the run is going to take. So in this case, it's only going to be a 20-second run. So we can now go ahead and check this box. The next thing to check for is that the input reagents are loaded, and I've prepared those earlier and loaded them onto the base of the instrument, so I can go ahead and check that. And the sample and waste containers are in place. So we have a tube on the front of the on the collection sample collection area, and that's where the cartridge is going to feed directly into. And we have a waste collection vessel towards the back of the instrument, and that's where the waste arm is going to divert the waste fluid. So I will go ahead and check this. Now, next we have a list of 5 statuses. So as we can see, no fluid is found in the tubing. The pressure sensor is okay. The cartridge is loaded. We now need to clamp the cartridge and close the lid. So I'm going to go ahead and close the lid. And we have a button on the screen to clamp the cartridge. So that's just the clamp arms come down to make sure that there's a good seal between the cartridge and the base plate, or the block over which the cartridge is sitting. So we are now ready to click formulate. So let's move over to the instrument again. Okay, so the first thing is our sippers come down into the bottles, and they come all the way down to find the bottom of the vessels that are being used. The pumps then start opening to draw the fluid into the fluid path, and it continues to do that until the whole fluid path is primed. So the fluid level, it's approximately halfway through the fluid path now, but it will, like I said, it continues to prime the system until the fluid is all the way up to the cartridge. So the system is now ready to start formulating. So the waste arm is out so that the initial fluid is being diverted to waste. And the flow is about to start now. So our fluid flow is coming out, and now our waste arm has retracted to allow the fluid be collected into the sample collection vessel. So our total flow rate here is 120 ml per minute, because we have 60 ml per minute for dilution or for reagent inputs, and then our dilution input is doing a 1-to-1. So it brings it up to 120 ml per minute. So now the software is indicating that the formulation has been a success. So we can go ahead and open the lid, and our sample is now ready for downstream processing. So that is pretty much a low volume blaze run complete. And before I move on to show you how to set up the external vessels, I'm going to purge the lines. So there's fluid in the fluid path right now. So before we go taking off any of the sippers in order to connect the Blaze Plus tubing, we will just perform a purge action. So again, I'll close the lid. And that's just going to empty any fluid that is in the fluid path. Okay, great. So now I'm going to go ahead and show you the part of the Blaze Plus tubing kit. So in order to connect the Blaze fluid path to external vessels, tubing is provided in the tubing kit. So there's both input and output tubing available. So the input tubing consists of a threaded adapter and lure adapters at the other end. So what we're going to do now is open up the lid, remove the zippers and connect the input tubing so we can go ahead and connect up the external vessels. So there we go, we've removed the first zipper, which is just a threaded connector. And we're replacing it with the threaded adapter on the tubing assembly. Now the Blaze Plus hardware kit comes with these clips, and these are here to protect, to allow us to insert the tubing, which offers a protection from any pinching of the tubing, and it also prevents the tubing from moving away too far from the instrument, so it kind of keeps everything secure. So we will do the same now for the center zipper. And lastly, I will do the install of the tubing assembly for the left, or the dilution zipper. Okay, so our tubing is now ready, and I'm going to go ahead and wheel in our bioprocess bags, which are on IV poles. And these bags come with lure connections on one of the outputs. So I'm going to go ahead and connect this bag directly up to the center. So I was just saying, we have our input vessels set up, and we are now ready to connect our output tubing and connect that to an output bioprocess bag. So I'm going to go ahead and connect the end that does not have a lure connection and attach it directly to the cartridge on the Blaze. First, I'm going to remove the sample, or the waste collector, because we are now bypassing that since we are connecting directly up to the cartridge. So there is a barb feature built onto the output of the Blaze cartridge, which allows for direct connection of the silicone tubing. Similar to the input tubing, the output tubing clips into the tube clips, which secures it in place, and it then diverts. There's from there's a valve that allows the user to manually divert some of the initial waste, or the initial fluid, to a waste vessel, and the other tube connects directly up to the lure connector on the output bag. And here we have it positioned directly under our Blaze instrument. So there is no longer a need to have a waste vessel on the instrument, so we can bring that down and feed in the waste side of the output tubing in there. And this, like I said, this valve is manually controlled by the end user. So the formulation, or the instrument, is now ready to formulate. So the next step is just to close the lid and set up the Blaze Plus recipe. So we move over to the Blaze software screen, and here we're going to create a new recipe again, and this time I'm going to call it Demo Blaze Plus. We're going to go ahead and choose the same cartridge, so the same cartridge can be used for high and low volumes. And we are going to input, this time, 10 litres of reagent volume. We'll maintain the same reagent ratio of 3:1, and we will input a flow rate of 115 ml per minute. So this time I want to draw your attention to the lower sipper checkboxes. Because we have connected directly up with the Blaze Plus tubing, we no longer need the sippers to come down to find our vessels. So it's important to uncheck these boxes. That's the way of letting the system know that we need increased priming because we're using a different input style. As we have bypassed the waste, we can set this to 0 and manually control that with the built-in valve on the tubing assembly. So this recipe is now ready to go. So I'll go ahead and click save. And as you can see on the recipe screen, we're getting somewhat of a warning saying not lowering sippers, but that's okay for Blaze Plus. It's just a way of indicating that this system is going to prime the lines as if external vessels are attached. And we are now ready to formulate again. And we're not going to do a Blaze Plus run. As you can see, this is going to take a little bit of time, close to an hour and a half to formulate 10 litres. But that is pretty much all the steps involved in getting the Blaze set up for a Blaze Plus or large batch run. So that's the end of the demo. Thank you very much for watching, and if you have any questions, please feel free to go ahead and ask Vicky or I. Thank you.